Hello, hello, hello. Welcome back to Let's Get Oral. I am so thrilled today to have my very prestigious friend here with us today, Dr. Freya, Marcelo, as I call him. We met years ago when he was still at Forsyth, but back then he was and still is a PhD in craniofacial molecular biology from UC system. And he's also a dentist, but periodontist in oral medicine and infectious disease from Harvard. Studied with the renowned scientists. I would say he is one of them at Forsyth, and now has a lab he maintains at JCVI in Southern California, but is also a new uh, coming back to the clinic. So thank you so much for being here today. Hi.
Hello. Thank you for having me. And you and Jonathan, I was just catching up with Jonathan's background and appreciate both of you inviting me here. I've spent like my whole life in this field, kind of wearing few different hats, but at the same time doing this goal of oral systemic relationships. I'm very excited to dive into all those worlds that possibly collide within the mouth. So let's get to oral.
Yes, let's get oral. It's I feel like it's been a miss not to have you on before now. So apologies and thank you for being here. I really wanted to touch base with you because I had just talked a lot about immunology and how it's affected by your mouth and periodontal disease. It can, give different phenotypes and then set you up for being more sick more often. But given that you had this really unique opportunity to evaluate salivary samples in the COVID population as it descended on us here on Earth, you have a really unique perspective. So just to backtrack, give us a kind of synopsis of your training, how you got here, and then moving into your COVID experience to kind of set the stage on your background and what you bring to the COVID experience, and then walk us through those salivary samples.
Oh, wow.
Yeah, I know
Well,
it's a big ask.
okay, I was born in. Well, it was never really a one light bulb moment, but rather really curiosity. Early in my career in Brazil, I'm from Rio de Janeiro. I seen and witnessed multiple people with oral lesions that were not explained properly. So when I was in dental school, I was second year dental school, I received a small little grant and I could bring populations to the school. I hired a bus driver. That was my research. I hired a bus driver and he would bring people from different communities and I would evaluate their oral health. What was interesting to me very early in my life is that suddenly I see people with periodontal abscess. And I'm like, well, can I see your blood work? Because in Brazil, we do have free medical care. And people were diabetic. And they told me, well, I never knew I was type 2 diabetic. And then because of evaluations of periodontal health and periodontal abscesses, we realized that a lot of people had that condition. And that was similar for lupus. And we saw multiple lesions that were not very common in oral health. And suddenly, those red lesions, those erythematous regions, were actually involved with SLE, systemic lupus. So, this was true for cardiovascular. The vasculature of the mouth is so fascinating because somebody's dying today having a heart attack. Emergency teams will give a sublingual drug, and we're still discussing the oral systemic. Oral is systemic and it's highly vascularized, and the nerves are in touch with everything, perception.
I just wanted to pause on that point just to double down on it.
Yeah,
So you're saying that during a heart attack, we can put sublingual what?
exactly. Sublingual medication will go straight
Uh-huh.
to the heart, and will control, somebody's heart rate immediately.
Right.
And that's what, medicine accepts. So through oral cavity, we can have access to the entire vasculature to the brain. And, so in your context of what you ask, I've seen clearly through my life from Brazil and then coming to the US in LA and then going to Harvard, I've seen this massive inflammatory burden. And doing my residency at Harvard, it was so fascinating being with all those professors that worked in microbiology and oral systemic conditions. It just brought the mechanism to the instinctive field that I always had on the sugarcane field. And the idea that there were names for those molecules and the interactors are quite specific. And so my career went from completely empirical but curiosity to then really scientific and detailed and really fascinated by the vasculature in the oral cavity, but also the immune response. While multiple people are fascinated by specialized T cells, B cells, which are so important for vaccines and different levels of immunization, I was more fascinated with other type of cells, which are the beginning of the response and how do they populate saliva in the oral cavity. So my career has been, I would not have changed a little bit, come from Brazil to the US and just every day diving into the oral immune host microbial world. So that has been, I did residency in periodontology. I've seen patients all my life. And now I see patients more frequently, but I run a laboratory at the J. Craig Venter Institute, and I'm an associate professor affiliated with University of California, San Diego. And I love that my career is sort of like very independent, gypsy, and it's not a formal academic process. And I love receiving students from all over the world and yeah, and being in some ways feeling successful because I have that mission every day, so that realization pushed me not just doing traditional clinical work but straight into oral systemic immunology.
I do remember when you got that job and how excited I was for you to make that transition. And then just shortly after, you obtained a pretty significant study, getting those salivary samples and really expanding what we know about now long COVID.
Exactly. And the idea of saliva and infectious disease, has been in the realm of craniofacial and dental research for a long time. But when you think of flu, you acquired via this oral, nasal, pharyngeal, oral portal. We're constantly interacting and not just releasing things, but acquiring things. So that's a big triangular portal here. So this oropharyngeal portal has a mucosal system, and a lot of medical doctors focus mostly on the nose area. And then dentists mostly are focused on operatory things, procedural things. It's almost like dentists are the orthopedic doctors for the mouth. It's just a next procedure.
That
But
is
there's
refreshing to hear you say it like that. Ugh.
Yes, yes. And they're thinking they can help just with procedure, but we can be investigative doctors. We can do medical dentistry, that dental medicine that's in fact investigative and investigate the diagnosis. Patients appreciate that so much. Through a really good diagnosis, through understanding more detail, we can tailor our treatments. That's the future. So during COVID, it was a big, first of all, my husband, who works with the biggest celebrities in the world, he was in China in 2019. And he comes back super sick, wearing a mask in December 2019. So he's basically case zero. And it was my 40th birthday in January in 2020. And I had a few friends, and a few people were wearing masks already. So that's January 5th, 2020. We were wearing masks and we were not understanding what was happening. He just told me this, what I'm having, something I've never had in my life. I'm like, wow, stay put, to have liquids and all those things. And I got it. So I go to the lab about January 5th, January 6th, and I'm having the highest fevers in the world. And I'm so excited for my lab, my work, my research. And beginning of the year, and I couldn't even move. I had no energy. I was like having hallucinations during the day, just by myself, suffering with that disease. And then eventually by February/March, we figure out that what was happening. And there was a moment that JCVI, the J. Craig Venter Institute in La Jolla, closed. Even though we are an infectious disease and genomic institute, we had to close just to understand what was happening. And I called Craig Venter myself and I said, hey, I'm gonna, I was already feeling better. I was having a lot of muscle pain and brain fog, but I didn't know exactly the mechanism. I told him, look, I am affiliated with San Diego. I'm in the infectious disease department. I'm calling David Smith, who is the chair, and I'm collecting matched blood and saliva samples. And at the time, it was acute samples, acute disease samples. But there was like, I could see immediately that we had time points. We had people that just had COVID for like 2, 3 weeks, some people that lingered for 30 days, some people that lingered for 90 days. So I was collecting samples and bringing my car, and I'm like, oh my gosh, should I wear like different masks in my car? You sort of
Wow.
feel lost. I was driving my samples. My technician wasn't coming because he said JCVI closed, so I'm not going to be there. And I can't force staff members to come in. So it was me and another PI at JCVI, Dr. Jean Tan, who's a virologist and specializes in flu. And we were pipetting the samples and doing ELISA. His ELISA protocol wasn't working for the saliva because saliva required a different sort of process. Blood, we were diluting like 1 to 50, but saliva, we required something different. And we were learning the protocols for those custom ELISAs that we were doing for SARS-CoV-2 antigens and antibodies. And it was all just happening, and it was just the willing and the curiosity. And very soon we found that there was a difference in severity and IgA responses. And IgA is right, it's really frequent for people that don't know in mucosal tissues, and it's a type of dimer antibody that defends mucosal systems like the mouth. And the idea was that this increased IgA and strong relationship of severity demonstrated that these molecules, in fact, impact immune cells like neutrophils to constantly be saturated and activated. And we knew that it was starting from the mouth, actually. In fact, the blood took a while to develop the entire cytokine panels that we then start to use inflammatory panel. So,
Wait, I'm going to
uh,
pause you. Hold on. So the IgA is your natural antibodies that you make in your mouth. It and part of how you defend against something like COVID. So when the creation of the IgA occurs that is specific towards SARS-CoV,
yeah,
then it saturates the immune cells in the mouth, like the neutrophil,
yes,
and
yes,
inspires
exactly.
an immune response. Is that what you're saying?
Yeah. So when you look closely in saliva, and we were doing studies early before the pandemics, that saliva had a lot of cells that were live. We know saliva is 99, according to the classic books. I don't think anybody revisited this percentage, but according to the most like well-known biochemical analysis, saliva is like 99% water, and it has enzymes, and it has cells, live cells, and it has a lot of proteins and swimming proteins like secretory IgA, which is our first line of mucosal response, which impact all this, opportunistic bacteria, fungi, viruses, and the entire oral microbiome. So it keeps that balance going. However, when you have SARS-CoV-2 active, replicating in the active phase, you start to have this huge IgA response, which wasn't, I don't know all the binding sites, but it wasn't that specific. It was really a strong amount of IgA. Through that, the neutrophil response, which we then did via proteomics, demonstrated that was highly correlated with the severe cases. So people that had mild to moderate had no severe neutrophil response. This is evaluating through like about 3 to 5,000 proteins via mass spectrometry of saliva. And we realized that the only people that had neutrophil dysfunctions with IgA increased amounts, just the amounts, demonstrated this really clear correlation of severity. So IgA neutrophil dysfunctions were highly correlated with severe. And so until today, there is no test for saliva that will account for those factors. So I believe that there is a point that dentistry needs to come up and say, okay, we can be diagnostics too. That's my hope, that we will get a paper like this that, by the way, took a while to publish. And I'm like, I told the editor, I'm like, you are just not understanding. This needs to be out there because people want to know about GA, want to know about proteomics and neutrophils. All these other little details don't matter. This is just a convenience cohort that we collected as we could and we published. So eventually they understood and they saw the dysfunction in saliva. And the idea that some aspects match with blood, because we had matched serum samples, and it was really cool. The first name of that paper was Jang, and Sunny was my postdoc at JCVS. She also had huge virology expertise before she came to my lab. So I was learning from her a lot. And Jang et al. Paper, she showed that there's some group of proteins that are inversely correlated with saliva. So not everything in blood correlates positively. In fact, there are other things that don't. Some cytokines, in fact, are completely opposite. And we're still learning why is that, why saliva is not a purified, more version of blood. And we are learning what is this inversion means and how can we create a mathematical correlation to understand how that relates to blood. We always do saliva and blood in our lab, or saliva, gingival fluid, and blood, because that's the gold standard, and we want to continue to show the importance of saliva, or variability of saliva according to the gold standard.
So to go back to some of your discovery, you mentioned that during the COVID infection, there's a bit of a delay between the production of antibodies and the cellular response. Can you talk a little bit about that?
Yeah, so there are two things that we have to clarify today. One is acute response, to a viral disease like SARS-CoV-2 infection. Another thing is a long-term response. Though, like let's say somebody has a flu and then suddenly they develop chronic bronchitis, or somebody will have HIV and then suddenly they develop something else. So all viral diseases have capability to linger, right? And then the name long COVID came up, where people start to develop these severe symptoms through time, and we were monitoring closely, not just saliva, but also tissue. So the excessive amount of IgA that we saw in acute response and neutrophil activation that was very dysfunctional, that we even don't know all the mechanism, we know some of the mechanism. We know that neutrophils are activated via NETosis, and NETosis, for people who don't know, it's neutrophil extracellular traps, and they release this intracellular DNA, and it's an evolutionary process to boom, let me control what's outside here. I'm a Spider-Man now. So neutrophils are Spider-Man, releasing all the web-like
I
stuff.
love it. Yes.
And the idea that they will do that, but we have millions and millions of neutrophils in saliva, and they releasing all this nets, it's not really great because it feeds forward more inflammation. So now we all know that saturation can also make you delay it later. Because if you're doing too much in the beginning, just go to the gym and do too much right in the first 15 minutes. You can't do it anymore after. And that works for the cells too. The excessive amount of migration from bone marrow to blood to then salivary glands and then coming through gingival fluid, all those cells, then later, they become dysfunctional and less activated. So the chronic phase, we start to then monitor saliva from, we had through a foundation called Polybio, which I'm on the board, and we started that foundation to start working with chronic conditions, not just SARS-CoV-2, Lyme and many other chronic conditions. I had this cohort from the UK and from Cardiff University with collaboration with David Price, MD there, and the hospital send us saliva and blood. But then we see a completely different profile. We see that there's in fact immunosuppression. So here I talked about an acute phase that show a lot of immune activation, but dysfunctional, not properly mounted to develop an innate transition to adaptive immunity. But now something that's very deficient, and through some proteomics that we did and cytokine analysis and antibody profiling, demonstrating that it doesn't mount a proper response. In fact, we found signals of immune suppression. So we have a excessive activation through then deficient. It's almost like HIV and AIDS, right? Which you have a lot of activation, and then if that individual will merge into AIDS, then there is immune suppression. Not the same way that the CD4, CD8 cells are depleted. Through other mechanisms, a lot of it is innate immunity.
Is this where you see that asymmetry of expression between the blood and the saliva?
Yes, completely inverse. The blood is activated dramatically in long COVID samples. Then saliva, it's not. Saliva actually has a huge immunosuppression. And and we don't know all the reasons.
Do you think it's compensatory?
It could be just feedback, but I think there are I suspect of other things, and I can give you some thoughts. Um, Through evolution, a lot of species, let's say like insects, right? They get contaminated with virus too, and ticks and mosquitoes. Their salivary glands are the places where they get a lot of infection. Part of it is viruses adapted to attach to epithelial cells. And these glands, salivary glands, are great place for that. And I believe that through replication, and we've seen this through other viruses like EBV or cytomegalovirus, when they're replicating, there is an environment of immune suppression surrounding the tissue. So we haven't shown this yet, but through your question, this great question, if it's just a feedback loop.
Do you think that the viral replication aspect is like self-supported and protecting by issuing out this program?
Yes, I think there's viral replication salivary glands of people that have long COVID. There is evidence already that people with long COVID have particles, have viral presence across multiple mucosal tissue. We just released a gut paper with spike proteins after 2 years of somebody getting infected. But the key is, how do we investigate what's next, right? And it's not just look at saliva, but we have to look at the tissue. Because this sentence I said, the virus replicating, probably creating immunosuppression, is a thought process, and it has been shown in other viruses, but it has not been shown for SARS-CoV-2 yet. And the way we're doing spatial transcriptomics. We localize regions within tissue, and then we get the gene or protein expression of the regions of the virus, the region of the person without the virus, and just compare those regions. So we can do 300 regions, let's say, from tissue, multiple regions, and assay around 18 to 20,000 genes at once. And through that, we UV cleavage of all those probes, you can get then the gene expression in situ. And this gives us the answer. And we are getting evidence towards this, what I'm saying. We believe.
Can you talk to me about all the sites, like oral, lung?
So we've done oral. We just published a bioarchive, first name Soria, Soria, and he works in the gut. There will be skin and there will be oral. And we are finding this again and again. If there's replication or activity or presence of spike protein or RNA scope, RNA, the region surrounding uh, the particle or the virus shows huge amount of immune suppression. And it could be through replication that will require other types of assays, but that could explain why saliva presents an immunosuppressive environment. It's not just inverted correlated with the blood. There is a real immunosuppression when we measure IL-13, IL-10, and all the specific anti-inflammatory proteins. They're up. They're up a lot. And that's not normal for multiple conditions that we've assayed cardiovascular before, saliva and blood. We've done type 2 diabetes, just surveying, what some reviewers think, oh, this is descriptive analysis. Well, it is, because to get to points of hypothesis, you have to build data banks to understand saliva and blood in multiple scenarios, type 2 diabetes, lupus, periodontal disease. And through those, I've seen some of these relationships. But this in SARS-CoV-2 is very specific, so it's not very general. And we're going to release the saliva and COVID and long COVID saliva and blood very soon. We did proteomics, cytokines, and antibody work. And this inverted relationship, I think, is fascinating. And I want people to, dive into our data, and we put all the raw data versus the analyzed data so people can try their own analysis and let us know. I think it's just fascinating too.
That's so interesting. So are you postulating potentially that the long COVID is virally replicating in the oral cavity and issuing that immune suppression locally?
Yeah. Yes, I am. And it might not be just salivary glands, minor salivary glands. It might be other epithelial regions. We see this in the gut right now. We tried both terminal colon and ileum, and we assayed a small population from Mount Sinai, collaboration with people there called David Petrino and Sorab, and they send all the samples to us. And we did the spatial transcriptomics assay that explained about the regions of interest and cleaving those regions for immune detection. It's real. It's real immune suppression every time it repeats. When in science there is replicability, you follow. You don't do the opposite. Just have a hypothesis and follow your hypothesis. No, when there's replicability and reliability, you follow that path. And I love that, training my students at JCVI and San Diego. I love to remind them this is a data that's being replicable. It's showing again and again. Let's follow this and keep going. And this
That's so
immune suppression is
exciting.
good.
Yeah, well, I guess it brings up a whole other ask, and that is like how in long COVID, what are the risk factors that we're talking about? I know that the research suggests that having periodontal disease would be a risk factor, but can you say more to this?
Oh yeah. So long COVID, it's a very complex condition. People mostly manifest their symptoms with brain fog as the number one. Out of all the epidemiological analysis, brain fog is the biggest one. Lack of, dysfunctional breathing. But then, if you have type 2 diabetes, any comorbidity that you had before will impact the severity of your symptoms of periodontal disease, because you're aspirating the amount of bacteria and you already have a chronic host microbial burden. Periodontal disease is quite severe for the blood. Research has shown that if you have active periodontal disease, you have an increased blood pro-inflammatory cytokines such as C-reactive protein, which is produced in the liver, and IL-1, interleukin-1. So any disposing condition will, but I was in this conference for Edge of Medicine, funded by Stephen Alexander Cohen Foundation, and they invite patients, scientists, and clinicians. And you do conference about chronic diseases such as long COVID, Lyme. We could see patients that were athletes in the past and they have long COVID, that they were speaking in the conference in a wheelchair. This is like very severe. And there is a huge amount of people with long COVID. However, funding is not being given. There's no funding today when I send grants for long COVID just to focus on the oral mucosa. It's very hard to get that funding because people don't understand the severity of long COVID. In some ways, to me, the story is repeating itself. It's like HIV and AIDS many years ago, and now COVID and long COVID. They send the patients to psychologists. They say that you're very stressed. So when I release this bioarchive and these papers that we are going to do now for long COVID within the polybio community, and encourage everybody to look into polybio.org, and all our symposiums are there recorded, the patients are craving for mechanism, they're craving for molecules. So when you ask these questions about, okay, we are just in the beginning, so predisposing factors are there. However, we're just in the beginning of the gene mechanism. We found genes around spike proteins. They're the same genes for colon cancer. We find genes that are upregulated in gut for Crohn's disease. So it's not like random, it's so relevant. And we, as we are doing this podcast, we call for private funding, we call for government funding, because long COVID needs attention for all mucosal tissues, including oral.
What is the current mode of treatment for these patients?
Okay, right now there's a lot of clinical trials being funded by PolyBio, which receive funding from private donors. And some treatments, clinical trials right now, are actually based on HIV trials. And they are using similar cocktails to see if people improve their outcomes. The hardest part is to really define those outcomes, because we don't have a quick test. Can you test a marker for long COVID before and a marker after? So they are basically doing symptoms, how the person feels and the symptoms, and that's still very subjective. But some of these clinical trials are succeeding. There's a professor in UW, he's more clinician, and he was doing a clinical trial for rapamycin for periodontal disease, low doses. And they are using that for long COVID. It was inspired by his clinical trial. And the longevity field are doing multiple clinical trials, and that's also helping the long COVID field. There are some promising factors, but there is nothing today that can clear spike protein in the guts. You go measure spike protein in gut of long COVID, it's packed. There's nothing today that can clear viral replication. If you use RNA-scope sense protein, you don't really find something that clear that. So number one, we need a little more of basic science before we do more and more and more of clinical trials. It's good to keep doing in tandem, but for me, a person that likes to procure and find mechanisms, we need a little more of basic science. We need to dissect the tissue more. We need to really understand. When I was in one polybio conference one time, this professor from UCSF, he was presenting about this radiographic staining that they do for MRI in multiple tissues. So they had this probe, and they can, because they find this staining for the MRI and the CT scan, they find the staining that's really high for certain levels of, I hope I'm not saying wrong, certain levels of immune cells. I believe those are types of CD8 cells. It could be a proxy for viral replication within tissues. So they get a full body image, um, and they get those, dark staining, radio opaque stainings across the body. And as he was showing, and he's showing the heart, he's showing lymph nodes, I'm looking at the craniofacial tissues and I'm like, oh my gosh, look at parotid glands. Parotid glands were like so stained in his, and he didn't even put in the paper. So I at the end I said, hey, can you move into the sections above by the craniofacial areas, by the orbital area? And as he was moving the slices, I said, well, those are parotid glands. That's what I suspect. Virus is replicating in those salivary glands. And then they put in the paper, that aspect. They didn't dive into it. It was just a description that they did. But I think we need more mechanism, Camille, to get to the right. So we need funding. The Recover initiative from NIH has been canceled for long COVID research. So we need actual direct funding for long COVID to work with craniofacial tissues as well as gut and lung and any other mucosal tissues.
I mean, it sounds like you have a really good opportunity as far as, creating a salivary test. If you have viral replication and, the cytokine profile to pull from, where can you push that in the private funding
We would
season?
like that. That I have delved into some entrepreneurship. I have some patents in the oral sensor technology, trying to do more live monitoring or even simple paper tests. At this moment, we would need really good funding to measure the population, not just small cohorts. I have really good samples in my lab. I have fridges and fridges and fridges of samples, whether it's saliva, matched blood, whether it's tissue. But we need some funding to monitor people through time and seeing which markers really pan out to continue.
Right.
And I think that's a hard work that needs to be done. I'm willing to do that, and we want to do that. We've been applying for that, but I think you're right. We should just uh, change the cards here and just do apply for phase one and phase two for private initiatives. And JCVI is very open now of being an incubator, and there are things happening now at our institute that we want to change our model versus a classic institution to a more modern environment.
Well, that's fantastic. We'll just pitch this podcast around and see what we can do for you.
Long COVID and saliva. Let's go. And helping those people, and having a marker that once they're doing a clinical trial, they can say that marker changed.
Yeah.
It's not just
They
a
don't need
one.
psychology, they need a treatment. Yeah.
They
And then
need,
I assume that was Dr. Jonathan Ann that you're talking about at University of Washington with the rampamycin work.
yeah,
Yeah. Are you
yeah.
working with him in any regard or?
No, we are not. But it was through the polybio connections
Ah, I
that we
see.
found his clinical trial, and then through Professor Akiko from Yale, who runs the infectious disease department there, and Emmy Pearl from Polybio, and David Petrino, they decided to do this clinical trial. I'm involved actually in the context of collecting salivas from the clinical trial and doing proteomics, mass spectrometry. Like, just to give you a hint, David Petrino is the leader there of kinesiology of Mount Sinai in New York, and he has several patients with long COVID coming up. And the idea of people saying, oh yeah, go do exercise, oh yeah, go to a psychologist, stop the stress. And people are like, they have mitochondrial dysfunction, they have muscle dysfunction, they
Right.
get sick after exercise. So it's not that easy to rehab those patients.
Right.
And Dr. Puccino has a incredible knowledge, has a book on a guide for long COVID treatment. And he sent me, maybe 120. Last year, I received this grant from Conrad Prebys Foundation. I always like to mention because my lab for 8 years, it's running through all these foundations, and this private funding from foundations, which is I'm very blessed and grateful. And they gave us funding to hire through the cancellation of DEI recruitment, VNH. We are hiring students from community college, and that's our grants that we wrote. So we're bringing community college students to JCVI. We interviewed 300 students last year and brought them, and one of them was the student Kashifa, and she was responsible for evaluating this clinical trial done in Mount Sinai. And they were basically doing a vagal nerve stimulation of people with long COVID disease. And we were doing just a saliva mass spectrometry. And it was a very good trial. They had sham treatment, they had real. We would need another podcast to explain just the trial. But in summary, we find that the vagal nerve stimulation with the device that they were using showed really decrease of the specific inflammatory components found in proteomics of saliva of these patients. It was like 5 visits through 12 weeks. They were monitoring, collecting saliva. So we can do like each individual, the population. We can do treatment versus sham. We can do visit 1 versus visit 5. And Kashifa just wrote now a book chapter first for Springer Nature. They invited us, and now she's writing this paper. She's a community college student. And this is only possible because years ago we received this funding for the human salivary proteome, the salivaryproteome.org, which is a free available resource for every database. The database that spoke about acute disease. Now we're going to put it the long COVID cohort, and then we're also going to put it there, the clinical trial, and many other labs have placed there. It's not just a repository. You can do analysis there, get your graphs, evaluate one patient versus the other, one population versus the other. And nobody requires informatics background. We made the tools for the clinician, for the biologist, for the curious minds. And salivaryproteome.org is the path of democratizing the data, and making the saliva proteomic data available to others.
That's incredible. I know that we're at time now, but I'll just be speechless for the rest of the day thinking about all the possibilities here. Um, I love to think that you inhabit all of the California coast at this point because you have now 3 different areas that you literally inhabit. And I just hope that your proximity to the San Francisco area will get you some, private funding for some of these ventures, because I think that they're so insanely important. And I'm just flabbergasted with the amount of work that you've been able to produce. It's incredible. Thank you for sharing.
Well, thank you both for inviting me. It's been such an amazing journey. I'm excited, I'm curious. I don't think there's ever a day I'm like, here we go again to the lab. No, it's fun. It's curiosity. And my team is young and very driven, and whatever gender, whatever background, we are open. We want curious minds, to be together and work in this mission. Whether it's the finding the mechanisms of the neutrophils, becoming the mitosis, or is the IgA inducing activation to the neutrophils, whether it's the proteomics, finding all the myeloperoxidase, those blood clotting proteins that impacts the disease. We think saliva is a portal for the body, and saliva is also a portal for non-invasive diagnosis. And
Yeah.
saliva is also underexplored because the medical community has not learned about saliva. They learn about stool, urine, and blood. And so I think I'm trying to simulate the next generation of dental professionals, hygienists in particular, clinicians, oral medicine, and perio, to investigate more saliva, and try to start doing tests and show the patients, some before and after. and start creating that philosophy and practice. And through time, we will find purpose that will change our treatments. But that's, we have to start the drive. We have to start doing it. So walk the walk, people. Come on.
Yes, yes, I'm in.
Well, I just want to say thank you. And yes, let's get oral, and let's get oral systemic, and oral is systemic. So we keep rolling.
Yes, yes.